Hello, this is Dr. Martin, and this section here is chapter 1.3 of chapter 1. This is the third series of lectures, and this is going to be a pretty brief lecture. I'm dropping this on Thursday of week 2. You will also have a quiz over chapter 1, and I'll drop that into the course probably Friday morning of week 2. Some of you didn't catch that on my intro video, but usually I'll give the quizzes a little later in the week, just to give you some time to walk through the material and walk through the lectures and making sure that you have access to all of your slides. But I'll drop that quiz in pretty quick. So here we are in chapter 1. Again, this is section 3. In the last section, chapter 2, we talked about taxonomy, developing a taxonomic system that names all living organisms. And it had to be a universal language. It had to be something not Chinese, not English, not Russian, not German, but it had to be based on a very universal language. And naming of organisms had to be the same name in China as it is in Brazil, as it is in the United States. Well, in this section here, we're going to talk about the scientific method, which is the title of this chapter. And again, we have to have a very standardized process of how we do science. It has to be the same in every country, in every discipline, in every part, every experience of science. And we call this the scientific method. It's a very standardized method, a series of steps in how we do science. There's a little bit of a cycle right here on this diagram here, but it talks about the steps of the interrelated steps of science. So let's walk through each of these just a little bit. You've heard about some of these in your high school and even in junior high science. You discussed some of these things. So first of all, in the scientific method, there's observations and there's questions. I would say the most widespread universal use of this that was exposed to the public was in 2020 when the coronavirus first began to show up in China, and it was named a pandemic about 30 days later, meaning it had basically encompassed the entire globe. That was a classic opportunity of observing and questioning things we experience in nature and send it through a series of steps so that we could try to find a solution. In this case, the best solution was a vaccine. Now, I know there was a lot of stuff going on in politics. We really politicized the vaccine. It was even on the presidential debates in 2020. And so anyway, whatever you feel about that is not, we want to relate it to science, not politics, and not in social media. So, but we know this, there was an extremely serious, ultimately killing, just in the state of Oklahoma, over 5,000 people died from the coronavirus, most of them over the age of 50, 55 or 60. We lost 5,000 people, 5,000 of our grandparents, parents, friends, people at work, just in the state of Oklahoma. So that was an issue. That was a problem. That was an observation. We began to question that. How can we resolve this? And it took a lot of money, a lot of time, and a lot of smart people to develop finally a pretty effective vaccine for the coronavirus. So that was the obvious observation, and it was the obvious question. And so then ultimately we develop hypothesis, and we kind of predict what the outcome of that hypothesis might be. A hypothesis is a kind of an upfront tentative explanation for what we are observing. It has to be testable. A hypothesis has to be testable, and it has to be testable in multiple areas by multiple different people. We call that repeatability. And then ultimately we develop some predictions based on the testing of that hypothesis. Ultimately, we develop an experiment where we gather data, we gather conclusions, The companies that developed the coronavirus vaccine, they had to prove these experiments worked. They had to give statistics on how effective they were, and they were about 93% effective early on in the vaccine process. That was impressive. And so we use data from these experiments, and then we have to show people outside of our experiment, outside of our sphere. We have to show them this data. It has to be very open, uh, very transparent, and that supports or doesn't support our hypothesis. Ultimately, we publish with data, with very clear presented data, and then that publishing that data is reviewed by people outside of us. We call that being peer reviewed. It's reviewed by our peers in our discipline. When I publish articles on bats, it first has to be reviewed by my peers. And by peers, we don't mean friends, acquaintances. We mean professionals within our discipline that can review my data, review my experiments, and then say, yes, this is credible. Yes, this can be published. Or no, this is not credible science. This is not a credible experience, and it cannot be published publicly. So it's very important that we understand what peer-reviewed means, and then we can publish our experiment in what we call a journal. So scientists test their hypothesis with the discovery in science in what we call very controlled experiments. Here's one here that's just used as an example, but we design experiments to test hypothesis in very now it's kind of nice if it's a laboratory experiment where we can do it in a petri dish, or we can do it using analysis, or use it using laboratory analytics. I'm an ecologist. My experiments are done in the field with critters, with animals that are hard to capture, and by me capturing and handling, it can cause problems. A lot of variability in the environment. So it just kind of depends on where that experiment is conducted. But we use experiments, we design experiments using what we call variables and controls. You've heard the term control before in your high school biology. There are variable aspects of an experiment, and then there are controlled aspects of an experiment. Here, let's talk about some variables. Remember this, it's kind of hard to remember this, and we have to remind ourselves the difference between an independent variable, or that's something that is in my experiment that is being manipulated, and then there are dependent variables. These are being measured, these dependent variables, and then there's standardized variables as well. So let's talk a little bit about this and see if we can understand the difference between a dependent variable and an independent variable. Here's a classic vaccine experiment. Notice there's two axes to this bar graph. There's the A side of the axis, and then the B, the bottom part of the axis. Usually when we do an axis like this, we do the independent variables across the bottom in a bar graph like this. These are, in this particular independent variable, these are doses to a vaccine. Different levels of doses. A placebo is a control. A placebo is where there's no drug, no impact. It's sometimes water, it's sometimes sugar. It's a placebo, it's a control. In other words, it's a standard that we are going to compare everything else to. Then we have a low and a medium and a high dose of this particular this experiment. This happens to be a rotavirus. Hey, one of the most serious viruses going on right now in the United States is the norovirus. It's a stomach virus, and it's being very widespread. I hope you don't have it or haven't had it. Somebody told me last week a student had the flu last week, and so, but the norovirus is probably the most serious virus in going across the United States and certain pockets of the United States right now. The RSV for infants is a very serious virus. So here in this experiment, we're giving different doses of the vaccine, a low, a medium, and a high, and then a no dose, and that's our control. Notice the effects. The seriousness of the virus is in blue. In the placebo, here's the virus that was in blue. It shows you, the strength of the virus or the illness. So across the bottom axis are the independent variables. These are the treatments. We give a low, a medium, high, but on the A side of the axis, these are the dependent variables. In other words, how much of the vaccine, or I'm sorry, how much the illness impacts a person is dependent on the level of the vaccine that was given. It's a dependent variable. The level of the vaccine given is an independent variable. It's a treatment. It's a difference. And that's kind of the difference between an independent variable and a dependent variable of a vaccine. So you can again see this here outlined in this experiment. Very classic experiment. So here's a question I want you to answer. You can answer this on your time. And remember, these PowerPoint slides are given in a different part of the module, but you can go through these PowerPoint slides without me talking on it. Ultimately, after experimental data has been collected, we develop a scientific theory, and this is an explanation. We use the data to support our theory. The theory has to be based on the data that was derived during the experiment. Sometimes it takes a long time. The record time of developing a vaccine for the coronavirus was remarkable. The virus, if you remember, in March 2020, by the end of 2020, we had doses of the vaccine. My mother developed the virus and ultimately died in January of 21, 1 year after it first hit the United States. And she was in a nursing home. She was in a nursing facility, but she was about a month away. Really, by the time she was hospitalized, only a couple of weeks away of having the vaccine, getting the vaccine. And now she had some other complications as well, but and that's nothing unique, because people who died of the virus as well. And so the record time by which that vaccine was developed was very remarkable, an incredible undertaking. But ultimately, we develop a theory based on the results of our experiment. Um, you know, Understanding the difference between a hypothesis and a theory and a fact, and sometimes in media, especially, these can be confused somewhat. Just to give you an example, Charles Darwin in his travels in the tropics, especially, he was from England and traveled around the world, but he noticed this flower right here. We have writings, his observations of this flower right here. He predicted that this flower right here, because of the way the tube of the flower and the reproductive parts of the flower were deep within this tube, these nectar tubes, he hypothesized that there had to be an insect or a bat or a bird or something that, in order to pollinate this flower and to propagate this flower, the pollinating animal had to have a very long tongue in order to do this. And so he developed a theory that is the case, that there had to be a pollinator out there that matched the shape of the nectar tube of this plant. And sure enough, years later, 30 years later, well after he was gone, a moth was found. Look at the tongue on this moth. But it was found that supported his theory that there was an animal with a very long tongue that would pollinate this plant. And hey, we've done the same thing in the southwestern United States. There's a pollinating bat with a very long, narrow snout, very long tongue, muscles on the side of the tongue that pushes that tongue out, and it's used to pollinate cacti in the southwestern United States and Mexico. The long-nosed bat. You can Google it. It's really a pretty cool story, but it's a pollinating bat. Many of the plants in the tropics in the desert are pollinated by bats. And so, sure enough, we found a bat with a long tongue so that its tongue gets down deep into the nectar tube of a particular flower of a cactus that the long-nosed bat pollinates. So those are theories that were ultimately supported by data and ultimately supported by um, experimental data. So data from experiments are interpreted more than one ways, but um, scientists tend to be very skeptical. Scientists, when it comes to experimentation, peer reviews, they're pretty skeptical, and that's a good thing, to be very skeptical in science so that hypotheses can be tested. So this is a section on the scientific method. You can kind of walk your way through it. Don't forget, there's going to be a quiz tomorrow. That's all on chapter 1, 3 lectures, section 1, section 2, section 3, 3 different lectures on chapter 1. Take that quiz tomorrow. Might even be another assignment that I drop into the course tomorrow. That's going to be kind of fun for you. So enjoy your day, enjoy your weekend, and let's get ready for week 3 next week.